You know, right now, deep inside you, there's this microscopic manager making life or death decisions trillions of times a second. It's wild to think about. Today, we're going to meet this ultimate cellular CEO, the one that really holds the key to life and, when things go wrong, to disease.
Okay, so picture this. Every single moment, trillions of cells in your body are doing this incredibly complex dance of division. It's how we grow, how we heal. But this dance, called mitosis, well, it's a super high-stakes operation. There is absolutely zero room for error.
I mean it. If just one cell messes up handling its genetic blueprint, the chromosomes, the consequences can be catastrophic. A single error isn't just a tiny mistake. It could be the very first step on the path to some really serious diseases, like cancer.
So the big question is, how on earth does the cell pull off this incredibly dangerous dance over and over without a hitch? Well, it relies on a master choreographer, a regulator that makes sure every single step is perfect. Let's meet it.
The hero of our story is this molecular machine called the anaphase promoting complex, or just APC/C for short. Now, its technical name, E3 ubiquitin ligase, sounds super complicated, but really it's just a fancy way of saying it's the cell's master scheduler and its demolition crew. It's the one making sure everything happens in the right order at exactly the right time. The best way to think about the APC/C is as the cell's ultimate quality control manager. It makes the critical call on the exact moment to move forward with division. It identifies the key proteins that are basically in the way, tags them for demolition, and just ensures the whole multi-stage process is orderly and incredibly precise.
So, how does this manager actually get the job done? It's not like it uses a wrecking ball or anything. No, it has a system that's way more sophisticated.
It runs this brilliant tag and demolish system called ubiquitination. So when a protein's job is done and it needs to go, the APC/C gets an order, and it literally slaps a small tag on it called ubiquitin. This tag is basically a demolition notice. The cell's recycling machinery spots the tag, and boom, it instantly destroys that protein, clearing the path for the next phase of division.
But here's the thing. This manager doesn't act alone, and that's crucial. For this level of precision, it relies on two different shift supervisors. They're called co-activators, who tell it exactly which proteins to tag and when. This division of labor is really the secret to its perfect timing.
Okay, so first up is the action supervisor, a molecule called CD20. It takes charge during the most dramatic phase, giving the order to tag proteins that are holding the chromosomes together. That one action triggers the physical separation of the chromosomes. Then, once that critical job is done, the second supervisor, CDH1, takes over the next shift. Think of CDH1 as the reset manager. It's in charge of the cleanup crew, making sure the cell exits division cleanly and gets totally ready for the next round.
And this just brilliantly shows the sheer elegance of this machine. The APCC is absolutely enormous, but a staggering 85% of its mass is just scaffolding. Its only purpose is to perfectly position the other 15%, the parts that actually do all the critical work. I mean, that's an incredible feat of natural engineering, right?
But what happens when things don't go according to plan? What if there's a problem? Well, the cell has a built-in emergency brake for the most critical moment of all. Let's see how it works.
So imagine this scene. The cell is right on the brink of pulling its chromosomes apart. The manager, with its supervisor CD20, is ready to give that final go signal. But then, what if the surveillance system spots a critical error? Like what if a chromosome is just not properly lined up?
This is where the cell's ultimate safety feature slams on the brakes, hard. It's called the Spindle Assembly Checkpoint, or SAC, and its entire job is to yell "stop" and halt everything until the problem is fixed.
So here's how it works. If even one chromosome is misaligned, it sends out an alarm. This alarm triggers the assembly of a stop signal, a little package called the mitotic checkpoint complex, or MCC. The MCC then physically races over and grabs on to the APC/C and its supervisor, CDC20, and completely shuts it down. It can't tag anything. The entire process of cell division is paused, just frozen in place, until every single chromosome is perfectly aligned and ready to go.
So you can see how this tiny manager's precision is absolutely essential for your health every single day. But what happens when this incredibly intricate system breaks down?
Yeah, a failure in this elegant system of checks and balances is incredibly dangerous. When the APC/C manager messes up, or its emergency brake doesn't work, the cell is sent down a terrifying path of genetic instability and chaos.
When the manager fails,
chromosomes just don't get divided correctly. This leads to a condition called chromosome instability, where cells end up with the wrong number of chromosomes. This state, which is known as aneuploidy, is a fundamental hallmark of almost all cancers. It's what drives tumor growth. And what's really wild is that a broken APC/C system can even help cancer cells adapt and become resistant to the drugs we designed to stop them from dividing.
So we're left with this really fascinating paradox, right? The APC/C is the cell's master of precision. It's a manager that exists to prevent chaos, and yet its own complexity makes it a critical point of failure. By understanding how this incredible machine works, and, just as importantly, how it breaks, we open a crucial window into the very nature of diseases like cancer, and hopefully how we might one day learn to fix them.